Ever wondered what makes you you? Why you have your mother's eyes or your grandfather's laugh? The answer lies in a tiny, elegant molecule, a twisted ladder that holds the blueprint for all living things. This is the story of the discovery of DNA. For centuries, humanity knew that traits were passed down, but how? The mechanism was a complete mystery. It was a puzzle that stumped the greatest minds. Scientists knew something inside our cells was carrying this information, but they couldn't see it, couldn't grasp it. Imagine trying to read a book written in a language you don't even know exists. That was the challenge. The race to crack this code heated up in the mid-20th century. Several brilliant scientists were on the hunt. In England, at King's College London, there was Maurice Wilkins and a gifted X-ray crystallographer named Rosalind Franklin. Franklin was a master of her craft, using X-rays to take pictures of molecules. Through painstaking work, she captured a stunningly clear image of the DNA molecule. This image, famously known as Photo 51, was a game-changer. It clearly hinted at a helical or spiral shape. Meanwhile, just a short train ride away at Cambridge University, two young, ambitious scientists were building physical models, trying to piece the puzzle together like a 3D jigsaw. They were the American James Watson and the British Francis Crick. They were brilliant, but they were missing a key piece of information. The story takes a pivotal turn when Maurice Wilkins, without Rosalind Franklin's knowledge or permission, showed Photo 51 to James Watson. For Watson, seeing that image was a eureka moment. The distinct X pattern in the photo screamed, "double helix." It was the confirmation he and Crick desperately needed. Armed with this crucial visual evidence and Franklin's detailed data on DNA's dimensions, Watson and Crick raced back to their lab. They realized that the rungs of the DNA ladder were made of pairs of chemicals called bases, adenine always pairing with thymine and guanine always pairing with cytosine. This pairing rule, known as base pairing, was the final key. It not only explained the structure, but also hinted at how DNA could copy itself so perfectly, by unzipping and using each strand as a template. In 1953, they published their findings in a short, one-page paper in the journal Nature. It was a monumental announcement that proposed the now iconic double helix structure of DNA. It was elegant, it was simple, and it explained everything. This discovery wasn't just an academic achievement, it unlocked the code of life itself. It paved the way for everything from genetic engineering and personalized medicine to forensic science and understanding our evolutionary history. We can now read our own genetic code, identify genetic diseases, and even develop therapies to correct them. Every time you hear about gene therapy, DNA testing, or the Human Genome Project, it all traces back to that groundbreaking moment in 1953. The discovery of the double helix is a powerful story of ambition, collaboration, and controversy. It reminds us that science is a human endeavor, a cumulative effort built on the work of many brilliant minds. While Watson, Crick, and Wilkins received the Nobel Prize in 1962 for their work, Rosalind Franklin had tragically passed away 4 years earlier, and Nobel Prizes are not awarded posthumously. Today, her critical contribution is widely recognized and celebrated. The story of DNA is the story of us. It's a twisting ladder of discovery that continues to climb, leading to a future we are only just beginning to imagine. Thank you so much for joining me on this journey into the code of life. If you found this fascinating, please hit that like button, subscribe for more stories from the world of science, and share this video with someone who loves a good discovery. See you in the next one.